I'm Laura Machesky, and yeah, so I'm honored and delighted to be here among these amazing women that have worked at the LMB. And my time at the LMB was 1993 to 1995. I was a postdoc with Rob Kay, and you can see him looking really nice and relaxed there, which is true. Rob is an amazing, friendly person to work with. And this was a time of lots of fun. So this is my first kind of experience of UK science. So I got used to doing some tea drinking in the afternoon, and I brought with me maybe my skills in making something that would not be allowed in the lab nowadays. We did some things that were maybe not as compliant with the current safety standards as they could have been. But I have had an amazing time, and it's influenced my career in multiple ways, including the amazing people that I've met here and the people that I'm still in touch with. And I can look out in the audience now and see lots of friends from the time when I was at the LMB. And it's great to hear how the science is still absolutely amazing at the LMB. So this is my kind of journey. I might as well put up. So I started at Johns Hopkins as a PhD student, and I went to the LMB on the advice of several of the faculty in the department who had actually done postdocs. So all those crazy Americans we heard about yesterday who come over and do a postdoc at the LMB because it's just a place that you have to go. And so that was my my start of my journey. And then from there, I moved from the LMB. I did some time in London with Alan Hall at the LMCB, a couple of 4 years there. And then I moved from there to Birmingham. And Birmingham was where I started really my independent group. And then, oh, sorry, I forgot to show. That's just the picture of what the LMB looked like, more or less, when I was there. And then from Birmingham, I went to a meeting in Glasgow, which was a kind of cancer research workshop. And I wasn't really even working on cancer at the time, but I saw, I also met amazing people there, and I saw some amazing science and also the beautiful hillsides of Glasgow. And I thought, okay, so Glasgow will be my next home. And I migrated there and have been there for 15 years and have just decided to come back to Cambridge. And so there's the Beatson in Glasgow, where I have been working up until 2 weeks ago. And here is the Department of Biochemistry, here where I've taken a chair. And I'm very excited to join back to Cambridge to the community. So I've been working for my whole career on actin dynamics and the idea of how a signal from outside the cell can trigger the actin to change in the cell, which can lead to changes in migration, changes in membrane trafficking, changes in the way the cell interacts with its environment. And this is the kind of picture that we had when I started my PhD. Some of this picture, I would say, still holds today, but a lot of it's changed. So the picture is roughly that calcium signaling was thought to trigger the uncapping of actin filaments, causing actin polymerization for cell migration, and that nucleators could regulate actin assembly in the cells. But what we didn't understand is what was wrong was that all the nucleators that we knew about seemed to be at the wrong end of the filament. So they were on the fast growing end. And that didn't really make a lot of sense, because you don't want to cap your fast growing end. You want to leave it free so you can nucleate filaments rapidly in response to a signal. And so then where my work came along was to find this kind of complex of proteins in acanthamoeba. So in Tom Pollard's lab, we worked on acanthamoeba, which didn't have any genetic powers, but was great for biochemistry. You could grow up masses of it, and you could purify proteins. So we actually did a lot of team science back in those days of purifying every actin binding protein that we possibly could on one big amoeba prep. And we would run these columns, and you would collect your own fractions off the bits that were yours, but everybody kind of mucked in together to make these preps. And one of the things that came out of this was this 7-protein complex, which we now know as ARP23, which we found was at the edges of the amoebas and near where the actin dynamics was all taking place. And that was exciting, because finally I convinced my supervisor to use peptide sequencing, which was expensive in those days, to try and identify these bands that we kept finding on the gel when we purified this complex. And it came out that those were the same as these yeast actin-like proteins that had just been identified in yeast at the time, but the function was unknown. And so we then hypothesized that we've got actin-like proteins that are in the right place where the dynamic actin is taking place. Maybe they're doing something. Maybe those are nucleators that could be regulating actin polymerization and might fill some of the gaps in our understanding in actin. And so this is Tom. He's currently, he's been at Yale for a lot of years after he moved from Johns Hopkins to a few other places as well. And he's now in California, where I think he's semi-retired. But we've had a lot of years together, really collaborating. And what happened at the same time, so this is now 1997, was that the pathogen world was actually contributing a lot to our understanding of actin dynamics, because these pathogens here, Listeria monocytogenes, they infect cells and they make these amazing actin tails and they can move around. And so between Matt Welsh's and Tim Mitchison's work and also Marie-France Carlier's work, we discovered that really the ARP2/3 complex was one of the major or the major nucleator of actin tails on Listeria. And so Matt actually called me up on the phone. At the time I was already in London. And he said, I found your complex from your paper in 1994, and we need to decide what to call it because it's, you know, it's nucleating actin. It probably is what you thought it was. And so I thought that was amazingly collegiate. We talked together and we probably didn't do a great job. ARP2/3 complex is not too catchy, but it is what it is. Lots of people don't like the name very much, but I've never been great at naming proteins. And then I went on to work together with my now husband, Robert Insel, and I was furiously, because I was now in Hall's lab and we were doing GTPase work at the time. And for small GTPases, the targets were still not known. It was known they were RAS-like proteins. They could make great things happen in cells when they were activated, including actin polymerization. And also, if you did the yeast 2 hybrid assay with GTPases, you were guaranteed loads of papers because you would find all these targets and then they would be new and you could connect them to all the signaling. So I instead did some 2 hybrid with ARP2/3 and in a kind of roundabout way hit the jackpot by finding that the WASP family proteins were binding to ARP2/3. And that was immediately apparent that was interesting because we had Pontus Aspenstrom in our lab at the time who was working on WASP, and he found that WASP was interacting with CDC42. And this kind of led to this hypothesis that these WASP family proteins provided an amazing connection between signals from G proteins, GTPases, tyrosine kinases towards activation of ARP2/3 that could give you a burst of actin in response to a signal. So that was a kind of, in a way, the birth of my independent career, is kind of hypothesizing this, and I've really been working in some form on this ever since. And that then was taken up by Tanya Svitkina and Gary Borsi, who discovered that when they looked with EM in the leading edges of cells, they discovered all this amazing branched actin. And that was something that we hadn't seen before and didn't know what it was about. But Tanya came up to me at a meeting. She said, oh, that ARP2/3 thing that you work on, I think maybe I see it in my cells. So can we have a look at, you know, do you have any antibodies so we can look at the branches and see if ARP2/3 is there? Well, they did find ARP2/3 at the branches. There was a lot of amazing controversy about whether the branches were there and what they were all about, but I think we now are pretty sure that they're there and they have ARP2/3. And then out of this came a kind of also, I would say, the most speedy paper that I've ever published, together with Tom Pollard and really Dyke Mullins and Laurent Blanchoin, and all of these people actually get a lot of credit for this, which was the idea that actin filaments plus ARP2/3 serve as the template for branched actin at the leading edge of the cell, and they catalyze this nucleation reaction. So the kind of dendritic nucleation hypothesis, which Tom has published, we have a figure in this paper showing, and he's published in a much more elegant way subsequently, but was the idea that this actin, ARP23, was really an actin brancher. And Robert Robinson and Roberto Dominguez have worked on the structure and really have now a nice model for how the filament, the mother filament, can bind to ARP23, and it can be activated to polymerize this new barbed end or new fast-growing end to make these branches that happen in lamellipodia. And now we have a very kind of big picture of in cells that ARP23 is the blue actin is the branched actin here, is doing a whole bunch of different processes. And we've identified loads of different activators, which are there in green, and inhibitors, which are there in red. From this nice review here from Roberto and Alexis Gautreau, is just showing the functions of branched actin are much more widespread than we maybe anticipated at the time. And then if I have another couple minutes, am I okay for time? Yeah. Just to tell you a quick news story, which is the idea of a new regulator of ARP23 based actin. So this is the structure of the wave or scarf complex, which is one of the actin nucleation promoting factors in the cell. And this structure was solved by Michael Rosen about 10 years ago. And it's known that this complex can be recruited to the plasma membrane. It binds to the small GTPase rack through a certain sequence that's conserved. And we found, together with Robert Insel and back to dictyostelium really, in a screen, sorry, I got ahead of myself. So this complex is known to activate ARP23 and cause branched actin. We found an inhibitor of this complex, which has homology to the CYFIP subunit of the complex, which is the rack binding subunit of the complex. And this inhibitor here competes for RAC1 with the complex and causes the complex to be more dynamic when it's active and recruited to the plasma membrane. And in mammals, there are two flavors of the CYRI protein. And we find that when we knock out CYRI, we have over recruitment of the ARP23 and the wave protein to the leading edge of the cell. So it kind of is a restrictor of the lamellipodia. But then a surprise came when my student could localize CYRI in cells. He found that CYRI-A, at least, can localize to these kind of cup-shaped structures in a very transient way. It flashes off and on. And we found that these cups are actually macropinosomes. And this is now back towards not only dictyostelium, and these are mammalian cells, but also Rob Kay's work, because he works on macropinocytosis and has done some beautiful work understanding the mechanisms of macropinocytosis. So our work led us to think about how cells use their actin to make lamellipodia and also macropinocytic cups. And so here we have the actin for migration, and in cancer, this can lead to invasion and metastasis, where macropinocytosis is known to be a fuel of tumor growth. And so we explored CYRI's role in macropinocytosis further, and we found that CYRI's recruited before Rab5 and actually after actin. So CYRI is a kind of at the point where the cup is closing in the macropinosome, and you can see there just some macropinosomes with dextran inside, with CYRI labeling them. So CYRI is found on those structures. And we found that integrins actually can traffic via these macropinosomes. And this can lead to changes in invasiveness and migration of cancer cells. So there's just CYRI-A and CYRI-B in different cells with the integrins in pink and the CYRI in blue, showing that they localize there. And that cells, when you knock out CYRI, are less able to do macropinocytosis. And this is just data here showing the lack of macropinocytosis with CYRI knockout, but they're actually in many cases become more migratory. So these are spider plots showing the tracks of cells over time, and this is the control, and these are the knockouts showing that they can be more migratory. So hence a kind of view of CYRI developing that it might regulate the balance between macropinocytosis and using your actin to eat versus using your actin to walk and invade and migrate. And so why do we care about macropinocytosis? Well, we talked about uptake of nutrients, but macropinocytosis is a way that the cells take things in and could also be used by cells for things like chemotherapy uptake and antigen presentation in the case of dendritic cells, which we heard about yesterday, and also the control of adhesion and signaling And this is the integrin uptake by macropinocytosis, which I think was a bit underappreciated. And this is just highlighting a couple of amazing papers from Daphne Barsagi's lab, who really put macropinocytosis into the cancer field. I have to thank all the talented people in my lab for their amazing work. I would say all of them contributed, but probably Ang Lee, my pointer is really not working, but Ang Lee and Heather Spence have been really instrumental in the macropinocytosis work. And then last bit is just to say, it is possible to do science and have a family. We've heard about that from a lot of you, and it helps if your husband is also one of your best collaborators. And thank you all.